Yogah Karmasu Kaushalam.
Good morning, all participants. Today the topic that is fermentation technology to produce biosimilars, a way towards making affordable drugs for the society. In fact, this topic I have selected uh, in two sessions. This one is the first session. Let me introduce myself. I am Dr. Rakesh Kumar R. Panchal, Associate Professor in the Department of Microbiology and Biotechnology, University School of Sciences, Gujarat University, Ahmedabad. As I say that this topic I have divided into two sessions, session 1, session 2. So we'll continue with first session, and then if time permits, then we may go for the another session. In fact, the speech is divided into major three parts. The first one is what we talk about, that is generics. Every day you heard something on generics. If you have any problem, maybe a kind of infection, disease, abnormality in your health, then straightway you go to doctor or physician, and physician always prescribe the medicine. While prescribing medicine, the physician used to go through your economic condition, and accordingly he will prescribe some of the medicines. There you also expect that the medicine must be generic. Where you go to pharmacist with the prescription, then you request pharmacist that if possible give me the generic medicine. So then question comes that what is this generic means? Another important point of discussion of this topic, that is where the chemotherapy is concerned, where the control of microorganism, that is infectious disease, that is concerned. This is caused by microbes. You will find that it was Paul Ehrlich who for the first time worked out the original research, you can say invention of the technology, and he for the first time has studied the syphilis caused by Treponema pallidum. And it was his idea that perhaps chemical agents may inhibit the growth of this Treponema pallidum or may kill this bacteria. With this concept, he had started trying numbers of chemical agents one after another, and to your surprise, he was failed once, twice, thrice, 4 times, 5 times, 6 times. Not like he had failed 100 times. He had tested numbers of agents, chemical agents. He had failed 200 times, 300 times, 400, 500 times. But still, the idea was healthy in his mind, and he was expecting that one or another day he will get this Treponema pallidum to be inhibited or be killed by chemical agent. So it was his 605 attempt, 605 attempt where he failed, and in his 606th attempt he got success. And surprisingly, it was an organic arsenic compound which killed Treponema pallidum in vitro. It was salvarsan which was been named as by him, and in fact it was been proved as magic bullet of the time, and the first person to open the doors of chemotherapy. the present day treatment, or what you call is the allopathic mode of treatment of disease, that is based upon by and large chemotherapy, chemical agents which use as therapeutic agents, isn't it? So by virtue of Paul Ehrlich, he is father of chemotherapy, and from then all the various chemical agents had been worked out to treat various kind of disease caused to human being. Later on, numbers of medicine came. These numbers of medicines may be chemical substance, so it may be synthesized by chemical reactions. And later on, biological molecules are also been entered there. It was 1929 where Alexander Fleming accidentally worked out a bacteria which was inhibited by something which was produced by fungi, Penicillium chrysogenum. In his plate, bacteria were there, and in his plate, by contamination, the penicillium has grown, and he observed that surrounding the colony of penicillium, there was no growth of bacteria. Then he had worked on this, and finally the new concept, that is the microorganism produce something which inhibit the growth of microorganisms. So it was being termed as antibiotic against the biological system, produced by the biological system. So one biological system produce something which inhibit the growth of other biological system. So that was another breakthrough in this treatment. An antibiotic that had proved when it was being worked out in 1929 by Alexander Fleming. Later on, during Second World War, numbers of wounded soldiers, and this wound goes for sepsis, and it was infection of staphylococci. There, people had used this antibiotic like anything, and it had proved to be excellent agent which prevent the sepsis among the soldiers. So thousands of soldiers' death that has been prevented, and recovery after the deep wounds. That was biologic. That was the biological molecule. So in my presentation, we will go through the technology, where I say that the first phase of this, that is the first lecture, where we will concentrate on the biologics, and in second phase we will go for biosimilars, right? So while studying biologics, we will say that initially where the definition of biotechnology had been introduced, people were working on biological system. Various microorganisms had been exploited, various other organisms, living creatures has been exploited, and people were trying to prove that this kind of technology is excellent, but it was not been authenticated. So United Nations and World Health Organization, they have accepted the definition of biotechnology. They have accepted the technology which has been termed as biotechnology in 1992 Convention on Biological Diversity. So they have gathered to discuss on diversity of biological system, and at that time they have defined biotechnology, that says that any technological application that uses biological system, particularly living organisms or derivative or thereof, to make or modify products and process for specific use. So that has been the wide definition of biotechnology. Says that this is a kind of technology where biological system has been used. Either biological system directly has been involved, or maybe the product of biological system is involved, and that kind of technology is been practiced as a biotechnology. Another definition comes, that is biopharmaceutical. Earlier it was a chemical agents which were being used as therapeutic agent. There are hundreds of different chemical substances which were being used as the agents to treat the diseases. But later on, as I say, that biological system has been involved, the product from biological system has came as a therapeutic agent, and there used to be another concept came, that was biopharmaceutical, a drug created by means of biotechnology, especially genetic engineering. Says that recombinant DNA technology, there used to be recombinant protein, particularly the recent days you go through, that is the monoclonal antibody, typically derived from living organisms. Anything which is derived from living organism, that is been used as drug, and the industry involved in production of this kind of material, that has been called as biopharmaceutical. When you say that it is derived from living organism, you will say that it may be animal cell, it may be bacteria, it may be virus, it may be fungi, or it may be yeast, from where the agent has been derived. Okay. There you can get the examples of therapeutic proteins like cytokine, hormones, clotting factors, all that is being produced using biological system. The best example of this is insulin in case of diabetes. Again, it is produced from biological system. DNA vaccines, and these days people are working on RNA vaccine also, isn't it? That the COVID days, you say that coronavirus, and it is RNA virus. Ultimately, it has ruined the world, and people are expecting the RNA vaccine eagerly. Of course, the trials are going on. Russia claims that they have produced this vaccine, and again it is being used, but still it is not been there in the major countries of the world. And lastly, we'll say that monoclonal antibodies. This is the antibody produced from a single clone. It is an excellent technology, recently been used. New experimental modalities are also a part of biopharmaceutical, where you can say that typically gene therapy, this is used to treat the genetic disorders. Stem cell therapy, again it is used to cure certain kind of uncurable problems, where the stem cell from the person has been derived, and then you grow it in controlled environment and go for the formation of some small molecules, one thing. And for the future, you can predict that yes, certain organs also can be formed using stem cell therapy. And the last one is RNA viruses. There is all kind of research on RNA viruses. That is all part of biopharmaceuticals. And where we say biopharmaceutical, it is nothing but medical drugs made through biotechnology. So you are producing drug and you are using biotechnology. Therapeutic or preventive medicines have been produced using recombinant DNA technology. That is also part of this. Where you say the example of this, the first and foremost example of biopharmaceutical, that is the humulin. It is human insulin which was been produced by recombinant DNA technology. What has been done there? There they have isolated the gene responsible for formation of insulin from the human pancreatic cell, beta cell of Langerhans, from human pancreatic, and then this gene has been added to a vector, a vehicle which carry this gene to bacteria. So actually human gene carried using a vector to bacteria, E. coli. So now this gene is transferred to E. coli, and then E. coli started expressing this gene, and the protein is being produced by E. coli, and that protein is exactly similar to the protein which is been called as insulin, which has been produced by the human pancreas. The same kind of protein is produced by E. coli in liquid medium, that is flask. That protein is purified, and this purified protein has been used as therapeutic agent there. So it is a part of biopharmaceutical, and it had helped the diabetic people. Like anything says that the insulin which was been very costly, that insulin may be available to each and every diabetic patient easily, and costing is also brought down. Okay. Where you say biologic medicines, they are currently prescribed to treat various kinds of diseases, different conditions. Few examples, if you see the blood conditions like leukopenia, neutropenia, and pancytopenia. Various cancers are being treated by biological medicine: colon cancer, breast cancer, blood cancer. Certain immune system disorders are also being treated by biological medicine, that is rheumatoid arthritis, psoriasis, Crohn's disease. These are immune system disorder that is also been treated by biological molecules. Neurological disorders, particularly multiple sclerosis, that is also been treated by biological molecules which are being produced in biopharmaceuticals. More than 400 biologics are in clinical trial. People are developing every day new and new biological molecule, go for clinical trial, prove it, and then get the license, get the agreement, and finally they start manufacturing and selling in market. These therapeutic molecules, they are used to treat various cancers, as you say, Alzheimer's disease, even heart disease. The biological molecules are used. Even in our laboratory also we are working on this kind of certain molecules. We'll come to this in the last phase of this lecture. Diabetes can be treated. HIV/AIDS can be treated. Autoimmune disorders are to be treated using the biological medicines. Then again question comes that what this biological medicine is. What are the biologics? Right? So we'll jump to this biologics. It represents fast growing segment of the pharmaceutical market. Biological molecules. You say that 58% products are in development pipeline. These are the products produced from biological system, and these are the products which are being used as therapeutic agent. Earlier it was only chemical molecules which were used as therapeutic agent. Later on the biological system has started. When the biological system has started, the scenario came at the stage where 58% of products that is in pipeline. More than 16% of market product, it is already there in market. Today if you go to market, more than 16% of product that is biological. If you find the growth of these biological molecules market, then the growth is very high. It says that 20% growth per year. Every year it is growing 20%, is highest. If you find the data of 2016, says that during 2016, end of 2016, if you go to market, pharmaceutical market, you will find that the total drug which has been sold, and if you compare only 10 top selling drugs, out of these 10 top selling drugs, the 7 drugs that were biologics. So now biologics are going to conquer the market of chemotherapeutic agents. This is having a potential to reach up to 50% of share of total pharmaceutical sale. Where we are talking in 16, it was 10 molecule out of top selling 10. 7 molecules were biologic. And these biologic molecule has potential to conquer 50% total market of the pharmaceutical products. So it's going to, or even it can rise more than 50% also. Then a question comes that what it is and how it is manufactured. Says that manufacturing biopharmaceutical required certain set of reactions, certain set of the protocol which is to be followed. So first what you do is you are going to develop the host. As I say that insulin is manufactured from the beta cells of Langerhans of pancreas, human pancreas. You just isolate beta cells. Then from beta cell you isolate DNA. From DNA you isolate the particular gene responsible for formation of this insulin. So what you do is identify the human DNA sequence for the desired protein. Example is your insulin, suppose. So you are going to isolate DNA from the Langerhans islets, beta cells. Then after from that DNA you find out the exact sequence which is responsible for formation of the protein, that is insulin. Okay, once that gene is there with you, you have separated the gene. Now you have to select the vehicle to transfer this gene from the gene which is isolated from human cell to bacteria E. coli. So of course the vector must be familiar to the recipient cell. So where you are transferring to E. coli, then vector must be familiar to E. coli. So you can select the plasmid of E. coli. So you just isolate the plasmid from E. coli, and then this plasmid, it is a circular double-stranded molecule of DNA. So you just break open it. So circular is converted into linear, and this two linear you add the sequence which is called gene, right? So insert the gene into the vector, and then after where you insert the gene into the vector, now this vector molecule is your recombinant molecule, or it is called as recombinant DNA. Now this recombinant DNA, that is engineered vector, you transfer it to the host cell. So insert the gene into the genome of the host. So this plasmid which is engineered, it enters in the E. coli cell, and then after entering inside the cell, then this plasmid is integrated with the chromosome of E. coli. And when it is integrated with the chromosome of E. coli, your gene of interest that become the part of genome of the bacteria E. coli. This is modified cell. You can call recombinant E. coli. This recombinant E. coli, it is going to produce the product of your interest. Exact DNA sequence and type of host cells are used that is going to reflect the characteristics of the product. So whatever product which is this E. coli is producing, so which gene is transferred and which bacteria is used. So these two things are very important. So different gene, the different organisms are to be selected. So characteristics of the product that widely depend upon these two factors. Okay, that is the gene which has been cloned, and another one is the host which is going to receive the gene. Okay, so this is biological system, and that is how there are a lot of problems coming. Major steps involved in this process, that is you have engineered your DNA, you have engineered your bacteria. So this bacteria is there with you. Now what to do with the bacteria? So modifying the selected cell, that is done by you. Now you are going to grow this cell in mass. So growing a cell line from the original modified cell. So you are growing it in mass. Okay, after growing it in mass, growing a large number of cells from this, maybe in liquid media, cultivating them, provide them the environment to produce your product, and then once that product is produced, you are separating that product, that's purifying that product, and finally that product is packed and sold as a drug. So these all steps are very difficult, and that is how we say that the biologics are little costly compared to chemical drug. Where we say that biologics are little costly compared to chemical drugs, again we are coming to find the difference between your chemical drug and the biological drug. Difference in manufacturing. So small molecule drugs are always been the chemical drug which are molecular weight is small. It is low molecular weight drug. Best example is your aspirin, 180 Dalton molecular weight is small molecule. So if you are going to prepare or manufacture a chemical drug molecule, what you do is you just add and mix the known chemical reagent in a series of controlled and predictable chemical reaction. Here the production technique is usually the same technique as for the innovator product. So the company which has manufactured for the first time, that company has prescribed the steps, chemicals, reagents, and in the same step you will have to do the reaction in your company. The production process is highly standardized. Contaminants, that is the disturbing molecules, are consistent and quantifiable. Means in this chemical reaction, what kind of problem comes? Suppose from water, suppose you are adding water. So from water some chloride may be there in water and that chloride is creating problem. So it is consistently been there. You can quantify it. You just find out what is the chloride level and then you remove chloride from water and then that water is used. There your problem is solved. So contaminants are consistent and quantifiable. And that is how the innovator company is producing the product. Same product can be manufactured by any company if you follow the steps. So suppose that innovator company has gone for patent, has manufactured and sold in market and earned money. But what happened when patent is expired? After expiry of patent, anybody can study that reaction, can manufacture it exactly same. Since it is exactly same molecule from the innovator molecule, all this kind of drug molecules are called as generic drug. Of course, the research is not to be done. Of course, the rigorous testing is not required. And that is how the production cost of this kind of generic drug, that is comparatively low. Comparatively low meaning very low. Hence, a drug which is patented one, chemical drug which is available at the rate of say one tablet costing 5 rupees, the generic drug may be the one tablet costing 50 paise or less. That is possible because the same thing, same molecule, no difference at all. If you go to biologic, what happened? This is not possible because in biological molecule, lot of variations are there. I have shown the comparison of the conventional drug, that is chemical drug, and the biological drug. So if you go through molecular size, you will say that the conventional drug is having 10 to 1000 Dalton molecular weight and biologics has 5000 to 2 lakh Dalton molecular weight. Even the structure is simple in case of conventional drug, chemical molecule, whereas structure is quite complex, complicated in case of the biologic drugs. Complexity is more. Of course, the impurities are more. Leachables are there. Byproducts are there because you are doing process where biological system is being involved. Clinical behavior is well understood in case of chemical drug, but clinical behavior is complicated. Mode of action is also complicated in case of biological molecule. Manufacturing process is simpler in case of chemical drug synthesis. It is chemical synthesis, so it is simple. And here it is specific and complex biological process, and hence it is quite difficult. Batch to batch variation, if you go through the chemical drug synthesis, batch to batch variation is negligible, is not there. Small to negligible variation. Whereas here in biological system, biological drug biological molecule is synthesized, we will find that no two batches are same. No two batches are identical. Says that batch to batch variation is very high. Manufacturing cost is also very high in case of biological molecules, whereas chemical molecules it is relatively low. Just for understanding of this complexity, I have shown here is aspirin, molecular weight 180 Dalton. Now you compare this aspirin with the smallest molecule of biological origin that has been used as drug, and you can say it was the first molecule which was been approved by government authority, insulin. So if you compare it with insulin, you will find that aspirin 180 Dalton molecular weight, your insulin is 5800 Dalton molecular weight. So molecular weight is very high. Okay. Again, insulin is compared with another complicated biological molecule, that is human growth hormone. If you go through, that is 22,000 Dalton. If you go through the GCSF, that is nothing but granulocyte colony stimulating factor. So this is the factor where the granular white blood cells are being developed from your bone marrow, and this factor is going to help the differentiation, going to help the growth of granulocyte. So that factor is used in various diseases. That is 18,800 Dalton molecular weight. So compared to aspirin 180 only Dalton, insulin 5,800, then GCSF 18,800, or human growth hormone 22,000, and the recent one is monoclonal antibody. So monoclonal antibody, a single antibody molecule, the molecular weight is 1,50,000 Dalton. Now you compare this 1,50,000 Dalton molecule with the 180 Dalton molecule. 180 Dalton and 1,50,000 Dalton. So complexity is very high. And that is how it is very difficult to manufacture. So biologics are always difficult to manufacture. Biologics are always very difficult molecule to produce, and hence biologics are always costly compared to the small molecule drug, chemical drug. Again, the complexity is to be understood by another example. As I say that, so far as complexity is concerned, your aspirin 180 Dalton molecular weight molecule that is having 21 atoms. It is made up of 21 atoms. So you can compare it with 20 pound weight of your bicycle. So you are manufacturing bicycle like product, right? How complex it is. Now it is compared with human growth hormone. As I say that 22,000 Dalton molecular weight, 180 to 22,000. Here it is having around 3,000 atoms, human growth hormone. Now it is comparable with 3,000 pound motor car. So you are manufacturing bicycle and you are manufacturing motor car. How complex these two? It is very difficult to manufacture motor car rather than a bicycle. Not only this, motor car will jump into monoclonal antibody, 1,50,000 Dalton molecular weight and 25,000 atoms are there in this monoclonal antibody. Single molecule of monoclonal antibody that is made up of 25,000 atoms. You can compare it with private jet, which is 25,000 pound weight, complex. So you are manufacturing bicycle, you are manufacturing motor car, and you are manufacturing jet plane. How complex it is. So now, is it comparable? One aspirin tablet is costing 50 paise and one monoclonal antibody molecule is costing hundreds of rupees. Why? Because it is quite complicated molecule. And when it is a quite complicated molecule, the manufacturing is also complicated. Where we say manufacturing is complicated, what can be done? Stages of product approval and commercialization. If you go for biological molecule, you will find that first you will have to develop the product development. Once product development is over, that product is produced by your biological system, then you need to go for scale up. From a small scale, maybe what we do in our laboratory, we are doing experiment with 100 ml liquid medium into 50 ml flask. From this 100 ml, we'll have to scale up to 1 liter. It is to be scaled up to 10 liter, then after 100 liter, then after 1000 liter, and then lakhs of liter. You need engineering batches also to be controlled. Okay, once the product is been produced, you need to go for the clinical trial. That has been done phase 1, phase 2 and phase 3. All clinical trial is a tedious job. As you know that these days the corona vaccine is manufactured by our Indian company, but it is not there in market. What is the reason? It is going through the clinical trial phase. It has to go through the phase, pass the phases, and then after it can get the approval. Okay, you need to validate everything, process validation of commercial batches, right? So when you go for clinical trial, what is there? So initially you prepare a product, biological molecule, and then you prove that yes, this product is effective. Okay, once that product is effective, what you do is you further go for it is testing. The first is preclinical trial. This preclinical trial that has been done on animals. So if your molecule passes this trial, then that drug is approved for testing in human. Where it is approved for testing in human, the human test is done in 3 phases. Phase 1, phase 2, phase 3. Phase 1 is 20 to 80 participants, volunteers are taken. Your molecule is given to 20 to 80 individuals, humans. They are volunteers. Of course, the payment is to be done to them. Okay, if your molecule passes from this 20 to 80 persons batch, then the next phase comes, phase 2, 100 to 300 participants, volunteers. It is to be injected, it is to be given to 100 to 300 people. As you know that on 15th of August, people were expecting that India may come up with the COVID vaccine and it is to be marketed, maybe a kind of approval will be given. But that is not given. Why? Because on that day it was 280 people from AIIMS Hospital, New Delhi. They have injected this vaccine to 280 volunteers in New Delhi. That was phase 2. It is 100 to 300 people. Now if it clears phase 2, then again it has to go to phase 3, where 1000 to 3000 participants are to be given or injected your medicine, biological molecule, any drug you say. And when this phase 3 trial is over, it is successful, your molecule is passing through phase 1, phase 2, phase 3, it cleared this phase, then the drug is submitted for FDA approval, Food and Drug Administration approval. So FDA review committee used to be there. They confirm the safety and effectiveness of the molecule, and then this FDA gives approval. So at that stage your drug is approved. And when your drug is approved, that is not the end. You have to go for phase 4. And this phase 4 you have to prove that thousands of people where this drug is given in various hospitals, various areas. And when it proves effective, no side effect, no any health hazard, then this drug may be marketed by you. So then this drug comes to the market. So it had to go through a stringent clinical trial. And then the drug come to the surface, come to the market. Okay. So when we talk about biopharmaceuticals, where we say that there is a strong relationship between manufacturing process of biopharmaceutical industry and the characteristics of the final product. So if the process is changed a little, the product will be changed. So far as biological system is involved, because biological system has to be grown under controlled environment, right? So manufacturing biologics is very complex process. A high level of precision is required to produce the consistent product time after time. Means every batch the same quality of product should be produced, and for that you need to go through high level of precision. Sorry, high level of precision, right? So now where you are going to pharmaceutical industry, we'll say that a small changes in the production process of this biological molecule production. Say for example, slight variation in the equipment which has been used, or slight changes in the environment where the product is formed. You will see that biopharmaceutical industries, they have controlled environment for the production. So slight variation, that is going to reflect very high in the product, right? So if you change the brand of equipment which is being used, again the product is changed. Environment, I say that temperature is fluctuated only 1 degree centigrade, again the product is changed. So what happens, that changes in the behavior of the cell which are manufacturing your biological molecule. So that cell behavior is changed, where the minor changes is there, right? So where the cell behavior is changed, cell will produce protein of slightly altered characters. So where protein of slightly altered character means what? Alteration in 3-dimensional structure of protein, alteration in acid-base variations. If the glycosylation is changed, you will find that your organism is producing slightly different character molecule. And when different character molecule, that is altered protein, that may have altered safety, that may have altered effectiveness. So effectiveness of protein is changed. The safe molecule protein may be changed to unsafe molecule. So again, you cannot sell it in market. So lot of variations are there, and that is how you need an expertise, microbiologists, biotechnologists, to work in the industry and to manufacture the product. And that is how the costing increases, and that is how the market rate is more of biological molecules. Now where the production is done, you will find that during the production cycle, you need to have certain tests which are mandatorily required. To assure high quality and consistency of final product, production process requires high level of monitoring and testing. During the process, monitoring and testing required. Now, if you compare this monitoring and testing of the production process of chemical drug and biological drug, there is wide variation. Because in biological drug manufacturing, around 250 in-process tests during the manufacturing is mandatorily be required. 250 different tests are to be done. And if you produce the chemical drug, in-process testing is around 50. So here you are doing 50 tests and producing the product. There you are doing 250 different tests and producing the product. And hence, again it is difficult to manufacture. The photograph which I have seen, that is the photo of the fermenter network in a biopharmaceutical industry. How complicated it is looking. These are the fermenters where you are using this fermenter and producing the product of your interest. What this fermenter is, says that it is nothing but a tank where the stirrer may be there, the temperature control is required, temperature, pH, dissolved oxygen, Everything is required to biological system. When you are growing biological system, it do require oxygen all the time, continuously. Right? So dissolved oxygen level should be monitored. It should not drop down. The pH, again, pH should not be fluctuated. So everything is to be monitored, right? So if you do this kind of thing, then you need fermenter, and the technology is called as fermentation technology. Where you need fermenter, it is nothing but a kind of machine which is used to produce, to nurture, to care, to grow microorganisms carefully. So this one is a picture which I have shown here. Says that the left side most, the rectangular unit is a control unit of fermenter. This one is a control unit of fermenter where everything is controlled from this. The second one where half glass and half stainless steel, cylindrical, and it is this one where you say stainless steel side, that is jacketed vessel. It is a jacketed vessel. This is called fermenter vessel. Of course, you will find a pressure gauge there. So what is pressure inside? That is also reflected. On top of this fermenter, you find a black colored motor. It is top driven fermenter. Right? Okay, there is a top French, that is stainless steel disc, circular. There are numbers of holes in this disc. Through these holes, numbers of sensors are being placed inside. Okay, bottom side, if you find, I say that it is jacketed stainless steel vessel, where you find 1, 2, 3 different probes which are connected. The first one, white colored wired probe, that is temperature probe, that detects the temperature, and that detection is given to the control unit, and control unit will take care about controlling the temperature automatically. We'll see it in next picture also. Okay, second one, black wired, little thin one, that is pH probe, that again detect which is the pH inside. It is also been transferred to the control unit, and control unit will take care about the pH inside. So how pH inside is been controlled? Says that there used to be peristaltic pump. In control unit, you will find that blue colored pump where various silicone tubes are connected. Right? So these pumps are peristaltic pump. That pump is operated by two way, one is manual and one is automatic. So in this pump, what happened? The tubes are connected, and that tubes are connected to the vessel, where one is pump which is connected with acid, sterilized acid. The other one is pump connected with sterilized alkali. So acid and alkali. Now it is PID control. So whatever pH probe gives the data, on the basis of this, the PID control will decide. Suppose your pH requirement is 6.5. So you set pH to 6.5. You ask this PID control to keep pH 6.5 inside the vessel. So how it has been controlled? Says that the peristaltic pumps are connected, and if pH is dropped by acid production inside by microorganism, then 6.5 pH is changing to 6.4, 6.3, 6.2. Immediately this PID control will on the alkali pump, and this peristaltic pump start rotating and add alkali in the vessel, so as your pH may slightly shoot up and comes to 6.5 again. Then this alkali pump stops. Now suppose alkali is produced inside, then it goes the other way, where the acid pump is started and acid is been added therein, so as to bring down the pH. So ultimately you need not to worry about, once you start fermenter, then automatically this machine is going to control the pH. So pH probe is there, probe will sense the pH, and then controller is going to control the pH. Okay, one more is the probe, that is dissolved oxygen probe, PO2 probe. So this detects the dissolved oxygen inside. That is also been controlled automatically. So when dissolved oxygen level drops down little bit, then the agitation speed increases or aeration increases. Here on control panel, right side you will find that blue color peristaltic pump 1, 2, 3, 4, where I say one is acid, one is alkali, then third one is anti-foam. So for anti-foam, suppose foaming is produced and foam reaches to the top flange, there one sensor is kept, that is called anti-foam sensor. The wire is connected with the control unit, so that will give signal to this as foam has touched the sensor. So immediately the PID control will start the anti-foam peristaltic pump, and anti-foam oil is dropped wise added in the vessel, so as foam may subside. Immediately when foam subsides, again signal of the anti-foam electrode stops, and this PID will stop the addition of anti-foam oil. So it is also controlled automatically, and fermentation run lasts for 7 days or 8 days, 10 days, whatever, depending upon product and organism manufacturing it. So till then the pH is controlled, till then the temperature is controlled, till then the anti-foam agent has been there, and the last peristaltic pump, fourth one, that is used for addition of the feeding material or nutrient addition. So if sugar is required at some time, then the pipe is connected, and one reservoir of sugar, sterilized liquid sugar solution, that has been kept therein, and this liquid sugar solution is been added as and when required. So there you may use the manual addition mode. The switch is there, and all switch can be kept it on auto and manual and off also, right? That can be. Okay, left side to this, that is to peristaltic pump, there is one transparent fiberglass unit, which is nothing but a rotameter. So your air inlet that is passing through this rotameter, and rotameter will get you the amount of air which has been added to the fermenter constantly. Volume of air per volume of medium, VVM, right? So that has been detected there. So that is the second one is your fermenter, okay, where all the sensors are connected, and it is complicated one, right? So this fermenter is used to be sterilized after every run. It is to be decant washed, and after washing the medium is charged, and this medium is to be sterilized. Entire fermenter along with medium is to be sterilized. For sterilization, the third unit, that is stainless steel cubicle unit, that is a boiler. Inside is the distilled water, normally we charge there inside, and then the heating coil is connected, and then it is also PID control, so temperature is to be kept, so water may boil, steam may be generated, of course pressure gauge is also there, and through stainless steel pipe this stream is allowed to pass inside the fermenter, right, constantly at the time of sterilization. Okay, once sterilization is over, again this boiler job is over. You can switch off this. Actually, this is also been controlled by your control unit. So control unit, you keep it on sterilize, and then just push it OK, so as the boiler may start, steam may be generated, it pass through this, and inside steam pressure comes to 15 pound, and then 121 degrees centigrade. It is kept for 30 minutes, and then after the steam is stopped, after it is allowed to cool down, and then after it can be inoculated. Okay, next to this boiler is another rectangle unit, that is called chiller. There also we used to take distilled water therein, and a refrigeration unit is connected, PID control, so temperature you can select, you can keep temperature whatever is required by you, and the chilled water through stainless steel pipe, it is circulated in the jacket. So this is used for temperature control. Again, it is been controlled through this control unit, right? So if my temperature requirement of the organism is 25 degrees centigrade, I am doing it in summer where the ambient temperature may be 40 degree, still my inside temperature remain 25, and chilled water of 25 degree is circulating throughout in jacket, and that is how the heat is generated inside by rotation, but still that heat can be taken care by this lower temperature water which is circulating there. So excellently designed instrument which is called fermenter, and the technology where you are using this fermenter, that is called fermentation technology. So what we do is again the top view of this fermenter, as I say that there is a pressure gauge there, and the topmost stainless steel cylinder like structure which you find that is in one SS pipe in between. This is nothing but a filter. This one is a steam filter, right? This kind of filter can be connected for air inlet also. Normally what we use is we use Teflon disposable filter. Many a time it is sterilizable also. So what we do is for some runs we can sterilize it again and use it again, reuse also. But by and large this kind of air filter are also been there. It may be 0.2, 0.4 micron porosity, right? So various valves are there, stainless steel connections are there in the fermenter, where you find that this is a bottom view where numbers of valves are connected. This is a bottom valve, drain valve, all the different valves, right? There used to be certain non-return valves are also there, right? And this kind of control valves are also there. It has been controlled through electric supply, that is otherwise called as solenoid valve. Okay, that is also been connected there. In as you see that the left side is your this thing sensors. Various sensors are connected to the bottom of this fermenter. Now when you go for one run of fermenter, after complete run you need to clean entire fermenter valve. Okay, once you clean entire fermenter, but you have to treat it like your baby, because it is very sensitive. So every valve you have to check personally, so as no leakage, sterility is to be maintained, and that is how a lot of things are to be worked out for this. So every batch your full attention is required. As you see that myself is working with some of the valve where some minor problem where I used to open it and then keep it okay, and then after I again seal it, and then after I check whether the problem is solved or not. Every batch you are supposed to check everything, right? So that is a tedious job. Okay. That is one laboratory fermenter. One more laboratory fermenter which is there in our laboratory, that is BioFlow 2000. It is made up of New Brunswick and this one is a 5 liter fermenter, right? 5 liter working volume. So in fact 5 liter working, you may say that 7 liter may be the total capacity. So this one is entire glass fermenter. It is made up of complete glass. Okay, and of course top driven. Control unit used to be there on top, and the addition unit, this is the probes connecting and controlling, right? So additional units are here. That you say that one is pH controller, one is the antifoam controller, right? For pH you need acid and alkali to be added, and that is how two peristaltic pumps are there for pH. Okay, for antifoam it is only one peristaltic pump which is very required, right? Temperature is controlled, right? For this particular fermenter, we do have one heating coil there on the bottom of this glass, where it is being controlled through this control unit, right? And right side yellow is marked, that is nothing but a rotameter, where the air is passing through this rotameter. Okay, so this kind of fermenter that is also there in our laboratory, where the closed view says that here all the probes are connected from top flange, because it is glass vessel and it is not being opened from bottom, right?
So it is nothing like a beaker like 7-liter unit, where inside is all been connected through a top flange. There is top stainless steel plate from where all this is pH electrode, then one is temperature electrode, then antifoam electrode, then dissolved oxygen electrode. This one is SS is dissolved oxygen electrode. So all electrodes are being connected from top, right? So this one is a control unit of Bioflow 2000 fermenter, and in this control unit you can increase or decrease whatever data. Here you need to feed the data, and then once you feed the data, you have to start the fermenter, and then entire control unit that is taking care of all temperature, all pH, and dissolved oxygen. Three things are being controlled by it. This is fine fermenter. Okay, as I said that for pH 2000 controller, that says that base and acid, these two peristaltic pump. Antifoam, there is one peristaltic pump. And for dissolved oxygen, it is not been controlled by this. It is been detected only, right? So no any other addition which is required. One more fermenter in our laboratory which we are working constantly with, that is 2-liter fermenter. This is Biostate B Light. This is from Sartorius company. This is USA made. Again, New Brunswick is also USA made. The first one which I have shown, that is a indigenous. That has been local made, Ahmedabad made fermenter. The second one, that is USA, New Brunswick. The third one is also USA, Sartorius. This is 2-liter fermenter. This fermenter is having additional advantage that says that in case of my previous fermenter, I say that this one is a glass vessel, but this is not jacketed vessel. So for control of temperature, I need to have one pipe which has been going inside, then running there, and then coming out. And through this pipe, I have to charge the chilled water that is running inside in the coil of this SS pipe. Whereas in case of this Sartorius fermenter, entire vessel, this is glass vessel, is jacketed one. So outside jacket, this two red color pipe is connected with chilled water supply. So from bottom chilled water in, and then it passes through, and from top chilled water out. This fermenter is having, again for pH control, you say that 2 peristaltic pump and then antifoam 1 peristaltic pump is required. Here no extra pump is there for feeding of the nutrient. But for that, what we do is we have a portable peristaltic pump purchased, and that has been kept along with this machine, so as we may use it, right? But this machine is always advantageous compared to your New Brunswick, because here in this machine we have facility of inlet of air, that is one thing, and second is inlet of oxygen. So directly we connect oxygen cylinder with it, and it is PID control, where the dissolved oxygen probe may sense the oxygen level there inside. With growth of microorganism, the oxygen level decreases, and when level decreases, what happens? It is to be increased. So it is increased by increase of the agitation. If agitation is increased, and if it is a fungi-like organism which is growing inside, then fungal mycelium may break, and when it breaks, again the viscosity problem is creating, because every fragment may be developing because nutrient is available. So that is a problem. So this machine, what it do? This machine will decide at its own at what time oxygen is to be supplied inside the machine, right? So inside the fermenter, it is connected with, one is atmospheric air, so air pump is connected, and second one is it is connected with oxygen cylinder. So PID control itself will decide at what time oxygen is to be pushed in. So your dissolved oxygen level will be maintained, and when dissolved oxygen level is maintained, your organism will maintain in healthy condition. That is how it will produce the product of your nutrition. Okay. So this is what the closer look of this New Brunswick fermenter. This is the top front of this New Brunswick. As I said that this one is pH electrode, this is temperature electrode, and this one is your I think this one is pH, and this is your dissolved oxygen, and this is temperature. Of course, small electrode is there for antifoam also. The top one black is a top driven motor, and then the RPM of motor, that is also been controlled by the
control unit only, right? Peristaltic pump, as I said, that alkali acid and then acid alkali and antifoam, these three only. All these switches are three-way switches. It is off, then on, and auto, three-way, right? All switch three-way. So you can select automatic function or maybe a manual function, right? This is connected with chiller. As I said, that chiller is also PID controlled, where the temperature of chiller, okay, my run temperature is 25 degrees, so I used to circulate 25-degree water to this. 25.6 is the temperature which is displayed therein. Constantly it circulates this 25-degree water in the jacket, and that is how the temperature of the unit that is being controlled. This one is an air pump which has been connected with my machine. As I say that constantly, this is just like as your air pump which you are using to charge air to your vehicles, right? That kind of small pumps, automatic one, that has been connected with, and constantly pressurized air is been passed through a rotameter, and rotameter is adjusted to particular volume of air per volume of medium, VVM, and then the constant supply of air. So constantly for 7 days your chiller should be on, your air pump should be on, and your controller should be on. Thereby all this process, if it runs well, then your batch will be okay. This is one more indigenous fermenter which we designed and then constructed through Ahmedabad agency. This is 20-liter fermenter, and it is a crude fermenter. In fact, there is a top driven motor, and then it is connected with a rotameter and air supply and air pump and other thing, and 20-liter working, so total around 28-liter carbo that has been used by us, and we are scale up process is done here up to 20 liters in our experiment. Okay, this one is the flask where we used to take a medium. To this medium we used to grow Aspergillus niger. This is one experimental detail I want to share with you. Says that this is a growth medium where we add the spores of Aspergillus niger. We used to count the spores, 10:6 spore per ml of this medium. We grow it there for 48 hours, where it forms very small, tiny spherical bits that is called pellets. So the pelletty growth is been obtained, and this pelletty growth we observe under microscope. This is closer look of pelletty growth. This is nothing but a small, tiny spherical bodies, just like as sand particles, like small spherical bodies. That is your pellet. So we collect some pellet on slide, put cover glass, and then observe under microscope. Where we observe under microscope, low power, single pellet is looking like this. This is a sphere of fungal mycelia. This one is Aspergillus. This is used for gluconic acid fermentation. Okay, so this is Aspergillus niger, and spherical body. In center it is dark, means packed mycelia, and periphery it is slightly fluffy one. Right. The same under microscope, low power, and it is been studied under high power objective. You will find that all the mycelia has been seen. Slightly greenish mycelia is observed. That says that it is having full of the cytoplasm therein, right? So excellently grown mycelia. From the structure and from the health of mycelia under microscope, we used to judge that now this is fully grown, so it is to be transferred to fermenter. So pelletty growth is observed in flask, and then this growth is transferred to fermenter, and the proportion is 15%. So what we do is, if you are taking 2 liters, then 15%, that says that 300 ml. So we used to transfer 300 ml of this kind of growth in this, and then after that maybe 0 minute, and then we start the fermenter, and all the parameters are controlled, and the manufacturing of a particular biologics that has been done. Again, this is a closer view of this pellet under microscope, right? Okay, so every time we have to observe under microscope, and then the decision is taken that this is the right time to transfer it to the production fermenter. When you are there in industry, when I was working with Alembic or Centrida Biochemicals, where we were manufacturing citric acid using the Aspergillus niger. In Alembic, we were manufacturing penicillin and erythromycin using a Penicillium chrysogenum fungi, that is penicillin, and then Micromona erythra,
this is the actinomycet where erythromycin was being manufactured. There we are having 2 lakh liter fermenter, very large one. Where you say 2 lakh liter fermenter means it is to be monitored. Everything is difficult. So all the time you have to monitor under microscope, and whatever decision you are taking, it goes to the 2 lakh liter fermenter, and if slight variation, again the batch may loss. 2 lakh liter material loss, if you calculate in money, it can be in 1 to 1.5 to 2.0 lakh rupees. So if your fungi or actinomycet is not obeying your order, not functioning well, then the company had to lose up to 2 lakh rupees per batch. 2 lakh liter, isn't it? It's great loss. And that this is how your duty is a crucial duty, being a microbiologist or biotechnologist, and everything rely upon your decision. So you have to develop high precision in all kind of functioning, right? So constantly you have to monitor everything. So far as manufacturing process is concerned, you'll find that cloning and selection of the organism is done by you. So you may develop organism well, you may transfer gene to the organism, go for recombinant DNA technology, and finally you will develop the organism. That organism is grown in mass. So you may have cell banking. That organism may be stored by you. So once you develop recombinant DNA technology and develop a recombinant organism, that organism is stored by you. Okay, and that stored organism is used frequently in batch by you, right? So there you need to control the warehouse also, that input of material and output of material, that is to be controlled. All quality control parameters are to be followed. A particular material is allowed to use because it is biological system. If you change the material, again your biological system changes. That is how it is very difficult. Manufacturing process, if you go through, you will find that there used to be upstream and downstream. Upstream, it is nothing but media preparation, inoculum preparation, cultivation of organism, production of your product is being done, and once product production that has been done there in the fermenter, then after the downstream starts. In downstream you have to go and recover your product, right? So for that, various strategy of chemical recovery and other thing that is been applied there. You may go for filtration, centrifugation, right? So various strategy is used. Then after, once the product is been separated, then after purification is done. Once it is purified, you may have a formula, so you may formulate it, and then again you finally filter it, and it may be 0.22 micron filter through which your product is passed. So that product is sterilized, and that sterilized product is injected in the human body, then it may not create any problem, right? Okay, that is downstream. And after downstream, the last stage, that is storage. Various biological molecule which has been produced, it is to be stored at lower temperature, right? So it is to be, means the care is to be taken that temperature should not be very high. Many product which we design, it may be the product which has not been altered at room temperature, and that is how it has been sold regularly in market. But there are certain products which are very sensitive for temperature. That product is used to be stored in refrigerator, and then it is to be kept in refrigerator. Various vaccines which we manufacture, certain kind of vaccine are to be kept at a lower temperature only, in refrigeration, maybe 4-degree temperature. And when you transport it, again it is to be transported in the air conditioned ones only, right? So storage is also a tedious job. So what you do is you may have filling, packaging, right, formulation, all these steps are to be done. And while filling, another thing, in next session I'll show you one small video of automatic robotic vial filling which has been done in industry, that I'll show you in next lecture. But everything is been done, finally sealing and labeling done, packaging and dispatch done. So till then you will have to be careful. But this is not the end. Where you are manufacturing the biological molecule, that lot of byproducts are there, lot of side products are there, and the effluent which has been generated, that is also very rich in nutrient. And when effluent is rich in nutrient, you need to handle the waste carefully. So waste handling and disposal is another headache for you. So you have to treat the waste material using some microorganism and bring down the BOD and COD level to permissible limit by which is set by the Pollution Control Board, and then you are allowed to dispose this waste material. So that is altogether a different task for you, and for that again it may be a 1 or 2 hours lecture which has been required for waste disposal. Means it is to be treatment, waste treatment and disposal, right? Okay, we'll skip at this time. Analytical microbiological testing, where I say that for biological molecule there are 250 various testing which is required. For chemical it may be 50 various testing required. This 250 various testing, all kind of test is done. In process sample testing, finished product testing, raw material testing, right? There you are going to test various things for say antimicrobial product. Then bioassay is to be done every 4 hours. We are doing bioassay in our laboratory, right? So numbers of testings are there. Data review is required. Release of data depends upon your testing, right? Again, the stability of product, that is also very important. Once the product is produced, once the product is ready, but how stable it is, how long it will last. So real-time stability is to be worked out in laboratory, and at the same time we are going for accelerated stability also. Says that that is going to decide the shelf life of your product. So your product is produced, your product is effective, but how long it remain effective? So that is also to be worked out by you in pharmaceutical industry. And then says that expiry date is been decided on the basis of accelerated stability testing, and that will going to decide the shelf life of product, and finally you go for dispatch to the market, where you are ready with the product to dispatch in the market. That you need to go through all validations. There used to be process validation, cleaning validation, software validation, equipment validation. Every equipment which you are using, it is to be validated. Instrument validation, method validation, which has been followed by you. Filter which you are using to filter the final product, again filter is to be validated. Whether that 0.22 micron filter which you are using, exactly it is allowing 0.22 micron molecule only to pass through, or maybe larger. That is also to be validated. So various validation strategies are to be followed, and you are there to go through, and all this documentation that is to be preserved batch wise. Every batch all the documents are generated, and that is to be used it, right? I'll give you one example and then we'll finish it. That Indian scenario of biologics, where we talk about biologics manufacturing in India. The Herceptin is a molecule, that is the active drug in Herceptin is trastuzumab. This molecule was the first biologic to be approved by Food and Drug Administration, which is used in certain breast cancer and stomach cancer. This molecule was been approved, and people manufactured it and selling there in the market. In next second phase of my lecture, we'll go and discuss about Herceptin, which has been manufactured by Roche USA. It's a very big multinational company, and that multinational company has gone through the patent of this Herceptin, and then after expiry of patent, numbers of company has jumped into and manufacture this molecule, and this was the first molecule which was similar biologics to be manufactured in India also. So Indian company manufacture the similar biologic also, first molecule here. This is Herceptin, and it was been allowed to approve market in United States also. So Indian company which has got approval to produce and market in India, and not only in India, to market in US also. So excellent molecule which was been there. Even today in 2020, if you talk about March 2020, there are numbers of company manufacturing Herceptin. This is similar biologics. Actually, it is biosimilars, right? So, after expiry of patent by the original manufacturer, any company can manufacture this molecule. That is nothing but biosimilar. Similar biological molecule. And similar biological molecule manufacturing cost is manifold less than the original innovator, because innovator has gone through all research. He had used money like anything and then develop a product, and that product is there in the market. So he must go for patent. He use the patent and sell the product in at very high rate in market for that many period of time. In India, where the patenting has been started and people are getting license for that, initially it was only 4 years where the Indian license was been given. Later on, it has been revised, even 10 years, 15 years, 20 years. US patent, if you go there, it is 20 years, where once you are getting patent, you have 20 years to manufacture it and sell it alone in the market and then earn money like any. But after expiry of patent, that molecule is open for manufacturing by any company, right? And then they people can just copy this molecule and then put it in market, so as the manufacturing cost is low, and when manufacturing cost is low, the market cost will also be low, and that is how the affordable drugs can be there after expiry of the patent by original manufacturer, original innovator, right? So this is what the scenario says, that Herceptin, and almost last slide, that says that the biotech drug approval 1982 to 2008. If you go through the first approval, that was came in 1982, only one molecule had been approved. Later on, if you go 1983, no molecule had been approved. It comes to 1984, no molecule. It comes to 1985, then one more molecule approved. Then 86, three molecule added to this. And likewise, if you go, then up to 1995, no more, very high number of molecules are not been approved, because numbers of companies are not coming forward. It's difficult to manufacture. Again, the patent has not been expired well, and that is how it was a problem. But when it came, the boon has been there in 1996. In 1 year only, 28 product has been approved, this biotech product, right? And then after, the figure is very high. 2008 it may be 32, but all these numbers, that is there on the basis of the trial which was been done, and this is that drug is approved for additional indications. If you go for a stringent indication, that the approval figure is 32 in 2008. If the general approval is been sought, then it can be 126 or even more, right? But the stringent protocol is followed, and this many products are been approved which are having practical value, and they have gone to the market directly, right? So that is, by and large, that is all about the chemical drug molecule and biological drug molecule which are being manufactured in India and maybe in abroad. But one thing is very clear, that chemical drug molecule manufacturing is comparatively simpler, and market cost is also comparatively low. But for the biological molecule, biologics, the manufacturing is quite complicated, and that is how the market rate is higher. If you go to purchase this kind of biologics, then of course you need to pay little more. And the company manufacturing this biological molecule for the first time, innovator company, they normally go for patent, and then throughout the patent period they are earning money, and when patent is expired, that molecule is open for any player to produce and market, and hence this molecule is being produced by other company than the original innovator. That molecule is called as the biosimilar or similar biologic, right? So in next lecture, we'll discuss on biosimilar or similar biologics in detail. Thank you all. Here is my
email and cell number. So all of you can note down this, and if you have any queries, questions, then you can drop email to my email ID. Thank you all.